Welcome back to my channel. The topic for today's presentation is Lac Operon. Before moving further in this presentation, I would highly recommend you to watch my previous video on Introduction to Operon in order to better understand this concept. Now, Lac Operon was discovered by Jacob and Monod in the genome of E.coli. This operon consists of following genes. First is a promoter region to which DNA-dependent RNA polymerase goes and binds. The operator region, which when switched on will result in the transcription of the genes in the operon and vice versa. A regulatory gene called I gene, which is the inhibitory gene, and three structural genes, namely Z, Y, and A. Z gene codes for an enzyme named beta-galactosidase, Y codes for the enzyme permease, whereas A codes for the enzyme transacetylase. Beta-galactosidase is the main enzyme of this operon which helps in the metabolism of lactose to glucose and galactose in order to derive energy out of lactose. Permease is the enzyme which permits the entry of lactose from the medium into the cell. And transacetylase is the enzyme which helps beta-galactosidase. Now the last gene in Lac Operon is the terminator, which terminates the transcription of its structural genes. Now, since this operon is regulated by an inhibitory gene which codes for a repressor protein, this operon is negatively regulated operon. Now case 1, in the absence of lactose. So if we have a petri plate in which we are growing or culturing E. coli, but this medium lacks lactose, then it is obvious that Lac Operon should not be functioning, right? Now these are the following steps. This is the arrangement of genes of Lac Operon, PIPOZYA. First P is the promoter of the I gene and the second P is the promoter of the structural genes. Now in the absence of lactose, I gene is transcribed to form repressor mRNA. This, upon translation, gives rise to a protein called repressor protein. This repressor protein goes and binds to the operator region in such a way that it inhibits the transcription of the structural genes. So as I said, in the absence of lactose, we do not need any enzyme that metabolizes lactose, right? Case 2, in the presence of lactose. Now in the presence of lactose also, I gene is transcribed and translated to give rise to repressor protein. However, now lactose from the medium enters into the cell and goes and binds to the repressor protein in such a way that it no longer goes and binds to the operator region since its configuration is changed. Now, since the operator region is switched on, Z, Y and A genes are transcribed into a single mRNA called Lac mRNA. This Lac mRNA is later on translated to form three enzymes named beta-galactosidase, permease and transacetylase from the genes Z, Y and A respectively. Beta-galactosidase, as I said, helps in the metabolism of lactose into glucose and galactose. Permease permits the entry of more lactose into the cell, and transacetylase helps beta-galactosidase. Now one thing to note here is that the I gene is expressed all the time constitutively in a Lac Operon. That is, both in case 1 and case 2. That is, in the absence or in the presence of lactose, I gene will be transcribed and translated to give rise to repressor protein. So this was the first operon to be discovered. Thank you. Please like, share and subscribe for more videos.